I'm Dr. Brian Crompton from the Dana-Farber Cancer Institute. I'm a pediatric oncologist who primarily takes care of patients with pediatric solid tumors, as well as children who have cancer predisposition syndromes that put them at increased risk for developing cancer during their lifetime. In my research laboratory, where I spend most of my time, we are using genomic approaches to improve the way we take care of patients with pediatric solid tumors. The primary way we do this is by the development of liquid biopsy technologies. The idea is that small fragments of tumor material can be found in the blood of patients who have cancer, and we can use blood samples to identify, detect, and profile these fragments of the tumor right from a blood sample to figure out whether patients are responding to therapy and whether they're likely to be cured by the therapy they're currently receiving. We're also trying to understand whether the changes that happen in cancer over time as a response to therapy can be detected in liquid biopsy to determine whether we can identify patterns of treatment resistance that may cause some tumors to be incompletely treated by the current therapies we use in clinic today. The funding generously provided by the Pediatric Cancer Research Foundation is helping us validate that these liquid biopsy assays are prognostic for patients with Ewing sarcoma, a very aggressive bone sarcoma that frequently occurs in children and adolescents. And with the funding from this work, we expect to be able to determine whether this assay can actually improve the way we care for patients with Ewing sarcoma in the future. A special thanks to all the patients who participate in clinical trials and biobanking efforts across the country. And thank you to the Pediatric Cancer Research Foundation and all the donors who contribute to the fundraising for their efforts. Without it, this type of work wouldn't be possible.